Hello everyone, my name is Aryan Raghuvanshi, pursuing chemistry honors from Kerodimal College, University of Delhi. My topic is Phenomenon of Inhibition, competitive, uncompetitive, non-competitive, including allosteric inhibition of enzyme. Inhibition. Enzymes are biological catalysts that speeds up chemical reaction in living cells. Enzyme inhibition refers to any process in which the activity of an enzyme is decreased or completely stopped by another molecule called an inhibitor. This inhibitor can resemble the substrate, can bind at the active site, or can bind at some other site on the enzyme and change its shape, which in turn affects how well the enzyme works. Enzyme inhibition can be broadly classified into two categories: reversible and irreversible inhibition. In irreversible inhibition, the inhibitor binds to the enzyme through weak, non-covalent interaction such as hydrogen bonds or ionic interactions, and it can dissociate easily. This means the enzyme activity can be restored when the inhibitor is removed. In irreversible inhibition, the inhibitor forms strong, usually covalent bond with the enzyme, often at or near the active site, and permanently inactivates it. However, in the presentation, the focus will mainly be on reversible types rather than irreversible ones. Now, let us look at the major reversible types of enzyme inhibition that will be discussed in detail. The four key reversible types are competitive inhibition, uncompetitive inhibition, non-competitive inhibition, and allosteric inhibition. Allosteric inhibition is a type of non-competitive inhibition. First, let us discuss competitive inhibition. In competitive inhibition, the inhibitor closely resembles the former substrate in structures and competes with the substrate for binding at the active site of the enzyme. Because the inhibitor substrate both try to occupy the same active site, only one of them can bind at a time, and the presence of the inhibitor reduces the number of active sites available for the substrate. This type of inhibition can often be overcome by increasing the concentration of the substrate, because higher substrate levels outcompete the inhibitor for the active site. Key features of competitive inhibition. First, in competitive inhibition, the inhibitor binds especially to the active site of the enzyme, directly blocking substrate binding. Second, the maximum velocity of the reaction, or Vmax, remains the same because it is sufficiently high concentration of substrate is present. It can displace the inhibitor and achieve the original maximum rate. However, the apparent Km, which is the Michaelis constant and reflects the affinity of the enzyme for the substrate, increases, indicating that a higher substrate concentration is needed to reach half of the Vmax. Example is ACE inhibitors. Now let us move to uncompetitive inhibition. In uncompetitive inhibition, the inhibitor does not bind to the free enzyme at all. Instead, it binds only to the enzyme substrate complex, often at a site that is created or unexposed after the substrate has bound. Because of this, uncompetitive inhibitors effectively lock the substrate in the enzyme substrate complex and prevent the reaction from proceeding to release the product. Uncompetitive inhibition has a very high characteristic effect on enzyme kinetics. The inhibitor binds at a site other than the active site, but the binding occurs only after the substrate is already bound, forming an enzyme substrate inhibitor complex. In this case, both Vmax and Km decreases because the enzyme substrate complex is trapped and cannot proceed efficiently to form product, and the apparent affinity for substrate seems higher since the ES complex is stabilized. One example is the action of lithium ions inhibiting the enzyme inositol monophosphatase, which is relevant in the pharmacology of mood disorders. Non-competitive inhibition. Now let us discuss non-competitive inhibition. In non-competitive inhibition, the inhibitor can bind either to the free enzyme or to the enzyme substrate complex, but it binds at a site different from the active site, often called an allosteric or regulatory site. When the inhibitor binds, it induces a conformational change in the enzyme that that reduces its catalytic efficiency. So even if the substrate is bound, the reaction proceeds more slowly or not at all. Now, key features of non-competitive inhibition. Here, the inhibitor does not prevent substrate binding directly, so the apparent Km remains essentially unchanged, indicating that the affinity of the enzyme for the substrate is not significantly altered. However, the maximum velocity Vmax decreases because a fraction of the enzyme molecules are effectively turned into less active or inactive forms by the inhibitor, reducing the total catalytic capacity. A classic example of non-competitive inhibition is the effect of certain heavy metals such as silver or mercury ions, which bind to the sulfidyl SH group in enzymes and distort their structure, thereby reducing their activity. Finally, let us look at allosteric inhibition, which is a type of non-competitive inhibition. In allosteric inhibition, the inhibitor binds to a specifically regulatory site on the enzyme known as an allosteric site, which is distinct from the active site. Binding at this allosteric site causes a conformational change in the enzyme that reduces the activity of the catalytic site, even though the active site itself may not be directly blocked. Key features of allosteric inhibition. Allosteric inhibition is most commonly observed in large multi-subunit enzymes where binding at one site can influence the behavior of other subunits. These enzymes often do not follow simple Michaelis-Menten kinetics. Instead, their velocity substrate concentration curve is sigmoidal or S-shaped, reflecting cooperative interaction between subunits. Allosteric inhibitors are frequently involved in feedback regulation, where the end product of a metabolic pathway binds to an enzyme early in the pathway to slow down further production and maintain balance. A well-known example is ATP acting as an allosteric inhibitor of the enzyme phosphofructokinase in glycolysis, allowing the cell to reduce the rate of glucose breakdown when the energy levels are already high. To summarize, enzyme inhibition is a fundamental concept in biochemistry that explains how enzyme activity can be controlled or reduced by different types of inhibitors: competitive, uncompetitive, non-competitive, and allosteric inhibition. Each involve different modes of binding and produce characteristics change in Vmax and Km as well as distinct kinetic patterns. Understanding these mechanisms is essential not only for grasping metabolic regulation in living cells but also for designing and using drugs that target specific enzymes and diseases. Thank you for watching this presentation on the phenomenon of enzyme inhibition. Thank you.